right, today we're going to be talking about chemical neurotransmitters in a little bit more detail. So neurons process and transmit information through both electrical and chemical signals, right? As a review, messages are transmitted down the axons as an electrical signal called the action potential, and action potentials reach the end of the axon presynaptic terminal, which is on the presynaptic side of a synapse. Action potentials cause vesicles to release neurotransmitter into the synaptic cleft, and those neurotransmitters bind to neurotransmitter receptors in the membrane of the postsynaptic cell. So, chemical transmission through the synapse can happen using endogenous substances or exogenous substances. An endogenous substance, that's an internal substance that's released by the presynaptic cell and binds to receptors on the postsynaptic cell. Those are your neurotransmitters that are made by your body. Whereas exogenous substances, the external substances that basically mimic neurotransmitters, things like cocaine and heroin, and they are, again, not made by your body, taken in some way, and they mimic neurotransmitters, they bind to receptors, and they have effects on your body and mind by working at the synapse. So those are the drugs, and we'll talk about drugs in the last part of this class particularly. But receptors recognize both neurotransmitters as well as their mimics, the drugs that we were talking about. All right, so both neurotransmitters work by being released into the synapse and binding to receptors, and drugs also work by being chemicals that are in the synapse that then bind to receptors as well. There are two kinds of neurotransmitter receptors on the postsynaptic membrane. There's ionotropic receptors and metabotropic. We'll start with the ionotropic receptors. Ionotropic receptors are simple. They are kind of look like this. There's a binding site, right? The neurotransmitter is released from a neuron, from another neuron. The neurotransmitter chemicals diffuse through the synapse, and here's a binding site, and that neurotransmitter can simply, and when there's no neurotransmitter there, the channel is closed, and then the neurotransmitter can bind to this receptor site, and the ion channel opens, and ions can flow through, again, whether that be sodium or chloride or something like that. Ionotropic receptors quickly change shape and open or close the ion channel when the neurotransmitter molecule binds to it. So it's as simple as that. The molecule binds, the channel opens, and ions can enter the cell. Metabotropic receptors don't contain ion channels. They're more complex. The neurotransmitter usually activates what we call a G protein, which then affects ion channels directly or indirectly through enzymes and second messengers. And so you can see here a simple ionotropic receptor where the neurotransmitter just binds and the channel opens. This is a metabotropic receptor where the neurotransmitter binds and through another protein called a G protein that can activate second messenger systems that then open ion channels in other parts of the membrane. So when those metabotropic receptors are activated, like we said, they activate second messengers that can change the excitability of a postsynaptic cell, make other slower changes to the cell, like changes in gene expression. So the receptor in a metabotropic receptor, or the receptor and the ion channel, are not a single protein. And they're called metabotropic because of the delayed movement of ions through the ion channel, and it requires metabolic steps, requires a number of steps. It's a much slower process, as you can see, because there's many more things that have to happen before that ion channel opens. So again, here's just a simple ionotropic receptor where the neurotransmitter binds, and then the ion channel opens, and the ions can flow through. Metabotropic, the neurotransmitter binds, a G protein is activated, and then they might activate, that G protein might activate second messengers that then open an ion channel. And last lecture, we talked about the fact we need to clean up the synapse. If you have extra neurotransmitter in the synapse, well, it can keep activating those same receptors. And basically, we want this very clean communication system. We want our neurons to have an action potential, say, I've got a message, send the message to the next neuron, and then reset. If you just have extra neurotransmitter floating around, then those extra neurotransmitters will start activating these receptors even when there's no message being sent. And that can be a problem. And that's actually how drugs work, which we'll talk about, that they may release extra neurotransmitters, so there's more receptors that are activated, or they may block the cleaning up process of the synapse. So again, those neurotransmitters stay in the synapse longer. So we need to clean up the synapse, and the neurotransmitters can either be broken down by an enzyme. We call that enzymatic deactivation, enzymatic degradation. Here's these enzymes, and they break up whatever neurotransmitter is left, right? And then those are basically waste products. Or there's these transporter proteins in the presynaptic cell that takes any extra neurotransmitter and brings it back into the presynaptic cell. And that process is called reuptake. So there are many different neurotransmitters. What is the criteria for a chemical to be a neurotransmitter? So number one, it has to be synthesized in the presynaptic neuron and stored in the axon terminal in a vesicle. It's released when the action potential reaches the axon terminal. It has to be recognized by receptors on the postsynaptic membrane. A neurotransmitter has to cause changes in a postsynaptic cell. And blocking its release interferes with a cell's ability to affect a postsynaptic cell. So there are many different types of neurotransmitters. Amino acid neurotransmitters, this would be something like GABA or glutamate. And amino acids are the basic building blocks of enzymes, hormones, and proteins. There are amine neurotransmitters like acetylcholine, dopamine, serotonin. These are basic nitrogen compounds. We have peptide neurotransmitters or neuropeptides like oxytocin and vasopressin. A peptide, right, is a compound made of 2 or more amino acids. And there are also gas neurotransmitters like nitric oxide and carbon monoxide. And you can see that there are all sorts of pathways in the brain that neurotransmitters and neurons are constantly sending messages and communicating with many, many parts of the brain. So we'll talk about amino acid neurotransmitters. The most common neurotransmitters in the brain are amino acid transmitters. Glutamate is one we'll talk quite a bit about. This is the most widespread excitatory neurotransmitter. It interacts with AMPA and NMDA receptors. AMPA receptors are the ionotropic, excitatory neurotransmitters. And NMDA receptors are ionotropic and excitatory. These are particularly used when we're making new memories, which we'll talk about in this class. And there are also some metabotropic glutamate receptors. Another amino acid neurotransmitter is GABA. We'll talk a lot about GABA as well. This is the most widespread inhibitory neurotransmitter. These interact with GABA-A receptors. Those are ionotropic. They're inhibitory and allow chloride ions to enter the cell. These GABA receptors, because they're inhibitory, right, allowing chloride to enter the cell, they actually work particularly in reducing anxiety. So we release GABA when we need to calm ourselves down, and many anti-anxiety drugs, something like Xanax, also causes an increase in GABA release, which then, because GABA is inhibitory, kind of calms the body down. Your heart rate slows down, your breathing slows down, the thoughts that maybe you're racing through someone's head start to slow down. Four important, so amine neurotransmitters, that's acetylcholine is a very important one. Acetylcholine plays a major role in transmission in the basal forebrain. So there are many cholinergic neurons that are found in the basal forebrain, the amygdala, the hippocampus. These are brain areas that we'll be talking about. Widespread loss of these neurons is associated with Alzheimer's disease. Disruption of the cholinergic pathways in rats interferes with learning and memory. So these are important for learning and memory. Other amine neurotransmitters, dopamine. Dopamine is important for many aspects of behavior. There's a mesostriatal pathway that's important for motor coordination that originates in the substantia nigra of the midbrain. Parkinson's disease results from a loss of these dopamine neurons in the substantia nigra, and Parkinson's is a motor disease where people have tremors and have difficulty with different types of movements. The mesolimbicortical pathway originates in the ventral tegmental area and is important in the reward system. When we talk about drug use, that's using dopamine in this reward system. And so that's part of what makes using drugs so rewarding, is the release of dopamine in this particular ventral tegmental area. So dopamine is very important, and we'll be talking quite a bit about it. Right? We've got the reward pathway that uses dopamine, as well as some motor pathways in the substantia nigra. Norepinephrine, that's another amine neurotransmitter that's important for control of many behaviors, from alertness, mood, sexual behavior, particularly important in the fight or flight response. Serotonin, another neurotransmitter that is important for many aspects of behavior like mood, vision, anxiety, etc. We'll talk a lot about serotonin because it appears to be low in people who are diagnosed with depression, and antidepressant drugs increase the neurotransmitter serotonin. Peptide neurotransmitters, there are opioid peptides. Opioid peptides mimic opiate drugs like morphine and basically reduce the perception of pain. One of these is called substance P. That's a peptide neurotransmitter that's very important in the perception of pain. So we'll talk about these types of neurotransmitters when we talk about sensation and pain. Gas peptides, a gas neurotransmitter, differs from other neurotransmitters in three ways. Number one is that it's produced outside of the axon terminals, mainly in the dendrites, and it actually diffuses out of the neuron as soon as it's produced. There are no receptors involved. It diffuses just directly into the target cell and activates second messengers. And it can also function as retrograde transmission by diffusing from the postsynaptic neuron back to the presynaptic neuron. We feel like this may be really important in memory formation because it changes the way the synapse functions. All right, thank you very much.